kidney, particularly the glomerulus, let's look at how urine is formed. And urine is going to be formed in a process that involves filtering out a lot of stuff from the blood, but returning most of it back to the blood, so that we only make about 1% of what we'll call the filtrate actually makes it to urine. So this begins in the glomerulus, passing through from the glomerular capillary. The filtrate goes into the capsule. Then it makes its way to the proximal tubule, the nephron loop, descending side, and then the nephron loop, the ascending side, and then moves to the distal tubule and the collecting duct. And within this region, urine is formed. So the filtrate is formed. Things that are needed to be brought back are brought back from the filtrate, and some things that needed to be added to the filtrate, even though they got through the filtration process, are added in that region. From there, we go to the minor calyx, which goes to a major calyx, which ends in the renal pelvis, down the ureter, through the urine to the urinary bladder, and then through the urethra to eliminate. So there are two types of nephrons in the kidneys. They are called cortical nephrons and juxtamedullary nephrons. The cortical nephrons are higher in the cortex. They're one short nephron loop. The majority of nephrons are cortical nephrons. They're called cortical nephrons because even their loop is mostly in the cortex. The juxtamedullary nephrons are lower in the cortex, and they have very long nephron loops. They're very important in regulating water balance and concentrating urine. They're only a small percentage of the nephrons, but they are an important part of the kidney structure. So those juxtamedullary apparatus is a structure that regulates the secretion of renin. The top portion of the ascending limb of the nephron loop for each nephron passes between the afferent and efferent arterioles. And at that point, the ascending limb comes in contact with the afferent arteriole and forms this juxtaglomerular, next to the glomerulus, apparatus. At that location, the cells of the structure are specialized. There are macula densa, which are tall, closely packed cells of the ascending limb, and the juxtaglomerular cells, which are large, vascular, smooth muscle cells of the afferent arteriole, shown here in this region. And renin would be secreted by the juxtaglomerular apparatus, and it works to regulate blood pressure by increasing blood pressure. So nephritis is an inflammation of the kidney. Glomerular nephritis is an inflammation within the glomerula. That can be an acute or a chronic condition. Acute glomerular nephritis is usually the result of an abnormal immune reaction. Happens about in some people 1 to 3 weeks after they've been infected with a type of streptococcus called beta hemolytic. The infection doesn't start in the kidney, but the antigen-antibody complexes form an insoluble substance that gets stuck in the glomerula, and that causes inflammatory response in those glomeruli and can severely damage the glomerula. There are a number of other autoimmune issues that lead to a similar condition. Chronic glomerulonephritis is a more progressive disease. More and more nephrons are damaged until kidney can't function. It's a prolonged inflammation, and it turns out that the fibrous tissue is going to replace the glomerular membranes, and fibrous tissue can't do what glomerular membranes do. So urine formation, the main function of the nephrons and the collecting ducts is to control the composition of body fluids and remove waste from the blood, and that's the production of urine. Urine is going to contain waste, excess water, and electrolytes. There are 3 parts to it: glomerular filtration, tubular reabsorption, and also tubular secretion. So we are going to make a filtrate. We're going to put almost everything into that filtrate from the blood that's in water. Then we're going to reabsorb most of it, because most of it we don't want to go away. But there are some things that didn't make it to the filtrate that we have to put in the filtrate, and that happens in tubular secretion. So we go from the afferent arteriole into the glomerular capillaries, where liquids are filtered out and filling the glomerular capsule. The blood continues down to the venule, and the filtered fluid called filtrate goes into the tubules, where some of it is sent back and other things are added to the urine. So the glomerular filtration is the first step of urine formation. Substances are moved from the blood into the glomerulus and the glomerular capsule. That would include water, small dissolved molecules, and ions. Larger molecules like proteins should remain in the blood. They're too large to pass through the pores. The glomerular capillaries are many times more permeable than other capillaries, but not enough to let proteins through, hopefully. They have structures on them called fenestrae or fenestrations. They are teeny openings in the walls, and this allows us to form what we'll call glomerular filtrate. It's the substance that we filter from the blood to the capsule, and it has about the same composition as interstitial fluid. So here we see it. The blood goes from the afferent arteriole to that structure, and then we see these podocytes that are part of that glomerular structure, and fluids make their way to this space in the capsule and then down the tube. So the main force that moves substances by filtration through the glomerular capillary wall is hydrostatic pressure of the blood inside the glomerulus. The afferent arteriole has a much larger diameter than the efferent arteriole. The resistance in the efferent arteriole increases the pressure within the glomerulus, and that favors filtration. So the net filtration pressure is going to be the force favoring filtration minus the force opposing filtration. So we've talked about favoring being the glomerular capillary hydrostatic pressure. The opposing forces are glomerular capillary osmotic pressure. Remember, those proteins are remaining in the blood. And the capsular hydrostatic pressure. So there's going to be some pressure within the capsule pushing back. Long as the net filtration in the glomerulus is positive, filtration will occur. And the glomerular filtration rate is directly proportional to the net filtration pressure. For the average adult, a glomerular filtration rate should be around 125 milliliters per minute or 180 liters per day. Blood plasma passes through about 60 times a day, but remember, only a small portion of that filtrate is actually going to be secreted as urine. GFR tends to remain relatively constant by a process known as autoregulation. There are conditions that increase or decrease the GFR within the limits in order to maintain that homeostasis. When GFR increases in cases of excess body fluid, the mechanisms responsible for keeping the GFR constant are autoregulation. Increased sympathetic impulses keep the GFR constant in changing blood pressure conditions by causing the afferent and the efferent arterioles to constrict simultaneously. The hormone-like renin-angiotensin system works to do that, and the hormonal atrial natriuretic peptide, AMP, and the ventricular natriuretic peptide, BNP. So the renin-angiotensin system responds to decreases in blood pressure. Then through vasoconstriction, secretion of ADH and aldosterone, that helps to conserve sodium and hydrogen ions, and that helps to increase blood pressure. It also helps to keep the GFR rate constant. I will not walk you through each one of these things in the table. In fact, down here it's kind of hard to read on this slide, but I do recommend that you go through these tables as you are reading. The tables are important. The tables and images and figures and a text are very important. So tubular reabsorption is the movement of substances from the renal tubules into the interstitial fluid, and where they diffuse into the peritubular capillaries. Most tubular reabsorption, about 70% of it, occurs in the proximal tubule, and that proximal tubule is lined with microvilli. Tubular tubular secretion, on the other hand, is the movement of waste from peritubular capillaries into the renal tubules. So tubular reabsorption. Different parts of the renal tubule reabsorb specific substances. Different substances are transported back into the internal environment by different methods of transport as well. So for active transport, we've got the movement of substance against their concentration gradient. There's a limited transport capacity because you can only transport if you have a receptor, and you can only use a receptor if it is not being occupied by something else. The renal plasma threshold is reached when there's more of a transported substance in the plasma than can be actively handled, and then it spills into the urine. And a good example of this is glucose in a diabetic patient. Osmosis is reabsorption of water, and endocytosis is a small protein reabsorption. So nephrotic syndrome is a set of symptoms that tend to occur in people with renal disease. Some renal disorders cause an increased permeability of glomerular membranes, which allows plasma proteins to enter the urine. The loss of those proteins in the urine causes widespread edema and an increased likelihood of infection. Decreased concentration of plasma proteins and decreased osmotic pressure leads to edema, ascites, which is fluid in the abdominal cavity, fluid accumulation in other body cavities. Edema and those other abnormal fluid accumulation leads to a decrease in blood volume and therefore pressure. And these events lead to the activation of the renin-angiotensin system and the production of ADH and aldosterone, which works to restore blood volume. So water reabsorption occurs passively, mainly in the proximal convoluted tubules. Ions are reabsorbed in the proximal tubule, and water is going to follow by the process of osmosis, giving you an isotonic filtrate at the end of the proximal tubule. Shown here. Tubular secretion. Substances are going to move from the plasma of the peritubular capillaries into the fluid of the renal tubules. Those substances are going to be waste that are destined to be secreted in urine. They have some active transport mechanisms that function in tubular secretion. They work in the opposite direction of tubular reabsorption. This would be the secretion of substances such as some drugs and ions, particularly potassium, and that's important for regulating hydrogen ions as well, important for regulating pH of body fluids. So here we see an example of tubular secretion. So hormones like aldosterone and cardiac natritic peptides are going to affect the concentration of urine, particularly the sodium concentration. So the ability of kidneys to maintain the internal environment depends mainly on their ability to concentrate urine by reabsorbing large volumes of water. In the distal tubule and the collecting ducts, which are impermeable to water if ADH is absent, water can be excreted as dilute urine. If ADH, antidiuretic hormone, is present, those segments become permeable and water is reabsorbed by osmosis into the extremely hypertonic medullary interstitial fluid. Urine becomes very concentrated under the direction of ADH, so ADH causes concentrated urine. Although the majority of the nephrons are cortical, the juxtamedullary neutrons are the most important, nephrons are the most important for regulating water reabsorption, which we can see with the difference between no ADH and high ADH. Again, I will not go through table 20.3. But as you are reading, as you are doing your homework, these are very important for you to work your way through. One of the things that makes the juxtamedullary nephrons important is the use of a countercurrent multiplier. That's a mechanism in the nephron loop that ensures the medullary interstitial fluid becomes hypertonic and concentrated urine is produced. It's known as a countercurrent multiplier. The vasa recta of the juxtamedullary nephrons contribute to the countercurrent mechanism. The thick ascending limb of the nephron loop is impermeable to water, but actively reabsorbs sodium. The descending nephron loop is permeable to water, but not the things dissolved in water. Since the thin descending loop is surrounded by hypertonic fluid, water leaves by osmosis and the fluid inside becomes hypertonic. Concentrated fluid would then move into the ascending loop. The sodium chloride is again actively reabsorbed, and as that process repeats, sodium chloride concentrations in the limb increases or multiplies. Concentration of fluid in both the tubular and interstitial fluid increases towards deeper portions of the renal medulla, which is why these need to have longer loops. So this one walks you through the countercurrent multiplier. If you ever known anything about a heat pump in a home, that is a countercurrent multiplier. And again, table 20.4 should walk you through each of those structures and how they work. So urea is a byproduct of amino acid catabolism. The plasma concentration reflects the amount of protein in a diet. It gets into the renal tubules through glomerular filtration, undergoes tubular reabsorption and then tubular secretion. About 80% of it is reabsorbed and about 20% is excreted in urine. The other nitrogen-containing waste is uric acid, and uric acid is a product of nucleic acid metabolism rather than protein. It enters the renal tubules through glomerular filtration. Active transport completely reabsorbs the filtered uric acid. About 10% of uric acid enters the urine through tubular secretion, and that is then released. So urine composition is going to reflect the volume of water and solutes the kidneys must eliminate from the body or retain to keep the internal environment in homeostasis. Composition of urine is going to vary from time to time due to dietary intake, but in general, it is about 95% water, usually going to contain metabolic waste products like urea, uric acid, and creatinine. Likely to contain trace amounts of amino acids and varying amounts of electrolytes. Typical urine volume is between 0.6 and 2.5 liters a day. 50 to 60 milliliters of urine output per hour is normal. The volume varies with fluid intake as well as environmental factors. Renal clearance is the rate at which a chemical is removed from the plasma by the kidneys. And this can be used to indicate the kidney's efficiency, damage to the glomerular, and progress of renal disease. There are a number of renal clearance tests. One uses inulin, which is a polysaccharide. Creatinine is a clearance test. These substances are both filtered by the glomerulus but not reabsorbed nor secreted, so it gives you a good rate by comparing what's in the blood to what's in the urine for the glomerular filtration rate. And I will stop there.